Forget the image of a scientist in a white lab coat frantically stirring beakers until their hands cramp. The future isn't about more human effort. It's about smarter delegation. Today, we're diving deep into the breakthrough where robotics and autonomous chemistry have finally outpaced traditional trial-and-error methods. We'll explore how a single workflow can generate 12 structurally diverse analogs from just 7 commercial precursors without a single human hand guiding the final decision. You'll see how flow chemistry is replacing glassware, why AI models are reproducing only 14% of what medicinal chemists design for some targets, but over 28% for others, and how this shift will revolutionize everything from malaria cures to cancer treatments. If you've ever wondered if the era of the human-driven lab is ending, this is where it begins. For decades, drug discovery has been a game of numbers and luck. Medicinal chemists spend their days running reactions like metal-catalyzed couplings or forming amide bonds, hoping 1 out of 50 attempts yields a molecule that actually works against a disease target. It's slow, it's expensive, and it relies heavily on human intuition, which varies from person to person. But the new paradigm changes everything by removing the guesswork entirely. Researchers have now developed autonomous synthetic workflows where the decision-maker is set with general success criteria at the very start of the process. Once that baseline is established, no human input is required to choose which samples get scaled up or which ones undergo subsequent diversification. This isn't just a minor efficiency upgrade. It's a fundamental shift in how we access chemical space. In one specific instance, this autonomous system accessed 12 different structurally diverse analogs based on only 7 commercially available precursors through 3 fully automated synthetic steps. Think about that ratio. Fewer starting materials yielding more complex variations without human intervention. The robot doesn't just guess. It yields the best reaction conditions and provides the exact settings needed for scale-up. This means pharmaceutical suppliers can move directly from a successful robotic run to production quantities, bypassing the months of validation usually required by human teams. The expertise driving this is flow chemistry, where systems of small, flexible tubes replace traditional beakers and flasks. This allows for precise control over reaction parameters that simply cannot be achieved in a standard flask, ensuring that the conditions discovered are robust enough for industrial manufacturing. The integration of artificial intelligence into this process reveals some fascinating nuances about how machines learn compared to humans. When fine-tuning these models to enrich libraries with molecules active toward specific biological targets, the results aren't uniform across all diseases. Against Staphylococcus aureus, a common bacteria, the AI model managed to reproduce 14% of the 6,051 holdout test molecules that medicinal chemists had previously designed. That sounds modest at first glance, but it represents a massive leap forward in efficiency when you consider how quickly an algorithm can screen thousands of options compared to a human team. However, the performance spikes significantly for other targets. Against Plasmodium falciparum, which causes malaria, the model reproduced 28% of the 1,240 test molecules designed by experts. Why such a difference? It suggests that the AI is particularly adept at navigating the complex chemical landscapes required to combat parasitic infections. Perhaps because the patterns in those active molecules are more distinct, or because the training data provided clearer signals for that specific biological interaction. This fine-tuning capability allows researchers to start with small sets of known active molecules and let the model extrapolate from there, effectively expanding the library of potential drugs without needing a vast initial dataset. It demonstrates that AI isn't just replacing human labor. It's learning to think in ways that complement human expertise by identifying patterns we might overlook in our fatigue-induced focus on routine reactions like metal-catalyzed couplings. Now, let's address the elephant in the room. What happens when the robot makes a choice a human would have rejected? In these autonomous workflows, the researchers intentionally stripped away the final veto power. They set general success criteria at the beginning, but beyond that point, there was zero human input regarding which samples were chosen for scale-up or subsequent diversification. This is radical because it forces the system to optimize purely based on the defined metrics rather than human preference or bias toward familiar chemical structures. Critics might argue that removing the human element risks generating molecules that are synthetically inaccessible or have poor stability profiles that a seasoned chemist would instinctively avoid. However, the data suggests otherwise. The workflow not only identifies promising leads, but also provides the specific settings required to scale them up. The robot yields the best reaction conditions, ensuring that what is discovered in the test tube can actually be produced in kilograms for clinical trials. This removes the valley of death, where many promising molecules fail simply because they were too difficult for a human team to synthesize efficiently at large scales. By relying on flow chemistry and autonomous decision-making, the system prioritizes manufacturability alongside biological activity. The result is a pipeline that doesn't just find active compounds. It finds compounds ready for the factory floor, fundamentally changing the economics of drug development by turning a multi-year process into something that can be iterated in days or weeks. So what does this mean for you, whether you're a student, an investor, or someone hoping for a new cure? It means the bottleneck of drug discovery is shifting from finding to validating. The ability to access 12 diverse analogs from 7 precursors via 3 autonomous steps drastically reduces the cost per lead generated. Pharmaceutical companies can now run hundreds of these autonomous experiments in parallel, rather than waiting months for one human team to complete a single synthesis series. The practical takeaway is that we are entering an era where the definition of medicinal chemist evolves from a reactor operator to a system architect. You won't be mixing chemicals. You will be programming the criteria, designing the flow chemistry networks, and interpreting the AI's selection logic. The technology has reached a point where it provides settings directly relevant for suppliers in the pharmaceutical industry. This isn't science fiction. It's happening right now with systems that can fine-tune models to reproduce nearly 30% of expert-designed molecules for complex targets like malaria. For investors, this opens up entirely new markets in automated lab infrastructure and AI-driven screening software. For patients, it accelerates the timeline from bench-side discovery to bedside treatment, turning what used to be a decade-long gamble into a calculated sprint toward cures. The age of the solitary genius staring at a bubbling flask is over, replaced by a symphony of robots and algorithms working in perfect harmony. We've seen how autonomous workflows can generate diverse chemical libraries without human interference, how AI models are outperforming humans on specific disease targets like malaria, and how flow chemistry bridges the gap between discovery and production. The implications for medicine are profound, potentially delivering new treatments for diseases that have stumped us for years in a fraction of the time and cost. If you're fascinated by where science is heading next, make sure to hit that subscribe button so you never miss an update on this rapid evolution. Drop a comment below. Do you think AI will eventually discover our first miracle drug, or does human intuition still hold the key? Let's discuss in the comments.